all for being here. I'm going to start talking about clinostats to simulate reduced gravity for low-cost space biology experiments. I know I'm going to talk about that because you enrolled here. So some of the topics that I'm going to be talking about in here is first, the space investigation problems that we have every time we need to or we want to make some space investigations, if it is, for example, under the biological part of space investigation. Then some of the solutions that we have come up to for this kind of problems that we have. Also, some of the Clinostat principles. I know that all of you are like very interested on space investigation, but in order to be everyone on the same page, I'm gonna be talking about some of the Clinostat principles. How do they work? What are the things that the clinostat make to some of the microorganisms that we're going to be using for the clinostat for clino-rotation. And one of the projects that I did in this scope, that is the development and verification of a low-budget clinostat built by undergraduate students. That is a presentation that Cole was talking about that I made in the AGICE the last year, and some of the implications that this development had, and also how the clinostats can be used in order to also simulate other orbits or other gravitational regimes. So first of all, some of the problems that we face every time we need or we're interested in making some space investigations are, for example, that they're pretty expensive. Even if you are in a university, if you are in a lab, if you are part of NASA, it is pretty expensive to make scientific research on the space field. It is because we need to get some things out of the Earth, or at least in the low orbit. So having these amounts of materials that we need and everything that we need to accomplish these kind of investigations, it makes this kind of research pretty expensive. So for example, the people that are in small labs or small teams, or even some universities in developing countries cannot afford this kind of investigation that we're talking about. So of course, there needs to be a cheaper option available to a bigger variety of scientists. And well, some of the solutions that we have come up to are some things called clinostats. But even clinostats and commercially available clinostats are too expensive for everyone to use them for some of their investigations. So, we faced that problem here in Guatemala when we were trying to start to make some biological space investigation. And we noticed that we were not going to be able to buy a clinostat. And we also noticed that we have the people and the knowledge to develop new tools for space investigation. And the only thing that we needed was guidance. So with this, I'm going to be talking about the clinostats and how we can be using that to generate a lot of more information on the space topic. So first of all, for the solutions that we have come up to is mainly to simulate some of the microgravity that microorganisms or even bigger organisms as humans, rats, or any other organism that you can think about feel when they're in the International Space Station, for example. And there are some things that we need to have in mind. That is that, for example, astronauts in the International Space Station don't feel zero gravity as it is mentioned in, for example, movies and stuff. But what they feel is a constant free fall because the International Space Station is falling constantly towards the Earth. So what we want to do is to simulate this constant free fall that the International Space Station, along with the astronauts and everything that is inside the International Space Station or even in the orbit, are experiencing when they are constant free falling towards the Earth. So one of the things that we have come up to for simulating these kind of situations that we can feel when we are at the lower orbit of Earth are parabolic flights that in bigger scope are planes that we are going to be having doing the trajectory of a roller coaster when we're going to be like this trajectory and then up and then down, and then I get this. And that is a constant thing that these planes do for a parabolic flight. And as you can see on your right, what it happens is that the plane is going to be in their horizontal trajectory, and they're going to be like in normal gravity. After that, they're going to be starting the parabolic trajectory, so they're going to start going up and up. And in that part of the trajectory, when they're going up, they're going to be experiencing a hypergravity situation where we're going to be having 1.8 gravities in there. And after that, we're going to start free falling again. And that is the space of time that we are going to be having hypergravity, weightlessness, or as we all know because of the movies, zero gravity. And after that, when the plane starts again getting its trajectory in a horizontal way, they're going to be experiencing again hypergravity of 1.8 gravities again. So the parabolic flight have 20 seconds of hypogravity or microgravity, and then 25 seconds of hypergravity. And we have 31 parabols for each flight, and it makes like 10 minutes of microgravity. So with this in mind, we know that some of the experiments that we want to do are not going to be able to be done because, for example, in microbiology, if we want to have bacteria there and we want to study, we will have the necessity of more than 10 minutes of microgravity, and it needs to be constant because bacteria are very sensitive to this kind of changes. So for example, they're going to be sensing when we are in hypogravity, when we are in hypergravity, and after that, all of that changes are not going to be like representable to the things that we want to accomplish for our biological experiments. So it only works for fast experiments or things that we can do like in this amount of time that we have. For example, the verification of some technology or experimentations that can be done like pretty fast. But for biological experiments, the using of this kind of microgravity simulations are not enough. So for this, we have small-scale microgravity simulators that are mainly three, the ones that we use commercially. One of them are Clinostats that simulate a constant freefall. The other ones are high aspect radio vessels that we also know as HARP. They also simulate a constant freefall. And we also have the random positioning machines, also known as RPMs, that what they simulate is, for example, that the bacteria or the cells that you're going to have in those experiments in the RPMs are not going to be able to know where up and down is. So they're not going to be able to have this gravitational orientation, and that is why we can say that we're simulating microgravity. So as you can see, clinostats and high aspect radio vessels use the same principle, the constant freefall. And that is why using both of them is, I don't know, is pretty important in the biological microgravity research. The only problem that we have in here is that, for example, clinostats have this process of verification of if it is really simulating microgravity or not. And something that clinostats have that high aspect radio vessels don't is that they have smaller containers. So they have like smaller radius where we are going to be having these particles in constant freefall. And high aspect radio vessels have this bigger radio of the hardware that they use. So some scientists say that maybe high aspect radio vessels are making some more particles to sediment because of this high radio that they are using. So clinostats are like a little better, but let's say it's like arbitrary and it's up to the opinion of every scientist. But I decided to use clinostats because of everything that I read and all the backstory that I had because of clinostats. So about the principles, let's imagine that you have a container, and into this container you have a liquid. And in this liquid, you're gonna suspend some particles. So if you leave the container like this, and you leave it stationary, then all the particles that you suspended in this single container are gonna start sedimenting and stay in the lower part of the container. If you start clino-rotating this, then the cells or the particles that you're gonna have suspended in this liquid are gonna start falling and falling and falling, but because of the viscosity and the liquid that we already have, and for example, also the movement of the liquid that we have inside here, the cells are gonna follow the trajectory of the laminar flow of the liquid. So they're gonna have this circular motion at the same time that they are falling. So if you can think about it, it sounds a lot like, for example, what they're feeling when they are in the International Space Station. They're constantly falling towards a direction, but they don't sediment. So to have better understanding of this, I made a video. So let's imagine that that black point is a particle that you have suspended in there. And the two other dotted lines are the security radio, that we call it like that, that is the place where the cells or the particles are gonna be moving. And we want that radio to be like pretty small or pretty specific, so the cells are not gonna be sedimenting or experiencing other type of motion that is not constant free fall. So that is why it is so important to have like materials that have a little radio and not, for example, like the high aspect ratio vessels. So we can have a small radio and a bigger radio of security of where we are gonna have the particles in constant free fall. So we choose Clinostats for these kind of developments and this kind of experiments. And because of that preference and all the things that I already knew about Clinostats, when I wanted to make some investigations here in Guatemala on biological research and space biology, I tried to make it with Clinostat. So that is how the development and verification of a low-budget clinostat built by undergraduate students in a developing country was born. So the team of the project of the creation of the micro-traveler, that is the name that we put to our clinostat, was fully made of Guatemalans, but one of us is in the United States at the Biosphere Space Technologies Agency and at the University of Colorado Boulder. So the story behind the experimental design and why we wanted to make our own clinostat is that on 2017, I wanted to make some investigations of Candida albicans and how it behaves under microgravity conditions. And because of the importance that it has, because it is a normal yeast that we have in our body. And for example, astronauts like experience bad things with Candida albicans in space because it starts being pathogenic and they have like this pretty bad rash in their whole body and even in their genitals. So it is pretty uncomfortable for astronauts to feel it on space. So I wanted to investigate how Candida albicans behaves under microgravity conditions, and specifically how could we inhibit this yeast growth. So I was starting with this experimental design, and I found out that it was pretty expensive to have my experiments flown to the International Space Station. So I started to look some analogs that we could use to make this kind of investigations. And with that, I found clinostats. And also I found the contact of my mentor that has been helping me with all this development among the years. And he told me that clinostats are the best, and we started to try to make our clinostats. So we started to look for how could we buy clinostats. We found out that it was pretty expensive too. So I asked myself, if I cannot buy one, then why not develop one? Of course, I'm not an engineer, I'm a biochemist and microbiologist, so I was not going to be able to develop the clinostat myself. But I have a pretty nice friend of mine that is called Freddy España. He's an engineer. And I asked him if he was okay with helping me with this development. And he was like, okay, let's do that. So we talked to Dr. Lucia, who is the one that mentored us in all this, in this travel and all this adventure. And he gave us some requirements that our clinostat needed to have. These requirements are based on logical things, and other things are based on how the clinostat that he used in bioserve space technologies for NASA-funded projects work. So first of all, we needed our clinostat to be able to fit within UVGIS, that is the university that I studied in, incubator. Also, we needed it to be powered via a regular wall outlet. So we needed to enable the use of 15 milliliters conical tubes, and that was pretty important because most of the clinostats that we can find commercially use specific hardware that they develop so they can use in their clinostats. So if, for example, we wanted to use the clinostat that they use in bioserve space technologies, we needed to buy a very specific fluid processing apparatus that they have that are like 15 milliliters conical tubes, but they are empty. And those are pretty expensive too. So we wanted to use some things that we could find in our lab without the need of buying new stuff. Also, it needed to be able to collocate the same amount of conical tubes on the same tray of the incubator, but for the static controls. Also, we needed to have the rotation of the conical tubes perpendicular to the gravity vector, and also a rotation of 8 to 10 RPMs, and that was before another investigation made about the ideal rotation speed for a clinostat that we later read about, and it was 5 RPM. So at the beginning, we thought that it was 8 to 10 RPMs, and then it was found that it is 5 RPMs, the ideal rotational speed for clino rotation. So the objectives of this first iteration of this first clinostat that we wanted to make was the development of a clinostat with the following characteristics. It needed to be low cost because only Freddie and I were gonna be working on it, so we don't have like that amount of money. Also, it needed to be easy to replicate because we wanted it to be like open source so everyone can use it. And also we want it to be verifiable, that it means that, for example, I am gonna be able to obtain the same results if I use a HARP, if I use an RPM, or if I send the things to the International Space Station. So with this, those are the objectives that we have, the general objectives. And also we wanted to open space investigation opportunities in Guatemala and more developing countries, and also increase space investigation possibilities. So this is how the first clinostat that we made looked. If you can see, it is only able to stand 6 samples at a time. It is 50 milliliters conical tube capability. It is something with, I don't know, it's pretty strange, but it worked. I made a verification test, and what I did so we could like be 100% sure that the things that we were looking at were the same as the other ones. I replicated already published work on morphology of Candida albicans under simulated microgravity using the HARP. And that paper was made by Altenborg, Nelson Price, and Heyman on 2008. So for example, you can see that I compare morphology in normal gravity and simulated microgravity. So for the normal gravity or terrestrial gravity, Earth gravity, we wanted the cells to look in their body form. That it means that we wanted them to be rod shape or circular, rounded, however you want to call it. And after that, in the simulated microgravity, we wanted the cells to look in their eiffel form, in their elongated form of the cell. So you can see that right here we have in the UVG is the results that I had with my own clinostat, that we had the same results for the morphology on the normal gravity cells that they had in the experiment that they did with the HARP. And also for the microgravity, we also had like these elongated cells in their Eiffel form that looked very alike the ones that they reported in their research. So that is how we made the first verification test. Only morphology comparisons were made. That's a problem. Also, we used simulated microgravity cultures in anaerobical way, sorry. And Candida albicans needs to be cultured under aerobic conditions. So it was a problem too. And we also were comparing to a HARP and not actual microgravity. So that was another problem. We made a second iteration, so we could have more than 6 samples. We were targeting to a total of 200 samples, but as you can see, it is pretty big. It didn't fit under the incubator that we were gonna be using. And also the vibrations that the motor did were too big, and the vibrations are something important for cells because it can cause stress on the cell, and with this stress, morphology changes. So as we were doing our verifications with morphology comparisons, we were not going to be able to compare if the, for example, elongation of the cells was being caused by the microgravity simulation or the shaking of the apparatus that we were using. So we didn't make any verification test for the second iteration, but we made a third one that it is the final microtraveler that we have, and it's the one that you can see in here. So in this one, we stopped using 15 milliliters conical tubes, as you can see. We started using Eppendorf tubes, and also almost all the parts that we were using were 3D printed or easy to have by hand. And I don't know, it was just easier to use this. And also something that changed is that we had the opportunity to see how an actual clinostat looked like in person. So that helped us to have a very better design of the clinostats. And we wanted this final clinostat to be open source so everyone could replicate it if they wanted to. So for example, we published what were the components that we were using to create this third iteration. Also, what was the code that we used for this clinostat to work under 5 RPMs, and also how did we cut the metal parts that we were using and how could they print the parts that were 3D printed. So if you're interested on replicating the microtraveler that we developed, these are the codes that you need to screenshot. The microtraveler code and designing and everything, you can find it in this QR code. And we are also developing a new open Clino that is going to be every single part of the Clino 3D printed. So you're not going to be like preoccupied of getting a laser cutter to get all the aluminum parts that we were using in the microtraveler. And we are going to be doing some upgrades to that. So if you want to be like up to date with the open Clino and what we're doing, you can also QR code, scan this QR code and enter to our mailing list so we can put you in contact with us every time we are going to be publishing the open Clino that we were developing. So for the third clinostat iteration, we also made a verification test comparing the morphology of the cells. And as you can see, again, in normal gravity, we have the cells in their budding form, the same as compared with the literature. And also under simulated microgravity, we have cells under their Eiffel form. And it's the same as we have in the literature. So again, the same challenges. We were only making morphology comparisons. Again, we were having these anaerobic conditions into the experiment that we were making. And as I told you, in this case, we were not using 50 milliliters conical tubes. Instead, we were using 1 milliliter microcentrifuge tubes. And again, we were comparing to a HARP and not actual microgravity. But the good thing that we have is that right now we're making some experiments so we can compare the same results that we had in here with actual microgravity results that some of the bioserv teams are working on. So I know I mentioned that clinostats are very expensive, and this is cheaper, but how cheaper it is, here you have the price of every unit and everything that we use for the development of this clinostat that we are presenting. And also we made these prices supposing that you don't have the access that we had, like for example to machine shops, or some of the components that we have were recycled from other experiments that we had already did. So supposing that you are starting from scratch and without any components of the ones that we were using, you're going to be investing $109.61 to create your own clinostat using the micro traveler design that we have. And comparing to commercially available clinostats, I'm not gonna mention the name of this company, but there is a commercial company that is renting clinostats, and if you pay monthly, you're gonna be like having to spend a lot of money. And if you want to have your own and you want to make one single payment, you're gonna be having to pay $22,000 to have your own clinostat. So if you compare that to our price is 0.5% the one payment. So it is pretty cheaper if you're gonna use our clinostat. So the implications of this clinostat is that we successfully developed a low budget and easy to replicate and also verifiable clinostat. We verify the conditions of simulated microgravity using morphological changes of CL beacons reported with a HARP, and also gravitational microbiology research is now possible in Guatemala, and more workshops focused on space sciences are being generated in the country, in part because of the clinostat. And also it can be used in other countries, small labs or small teams, as I mentioned later. So other research that I have done on clinostats and how it works with simulated microgravity, I'm gonna just be presenting the simulated microlunar and microgravity microbial research project that I worked on. It is a NASA-funded project, and it is again from Louisiana, the University of Colorado Boulder, and Bioserv Space Technologies. And the objectives of these specific experiments are of looking for the morphological changes and different dynamics of the cellular growth and aggregation of different bacterial and yeast cells, and also using different simulated gravitational regimes as microgravity, that is the one that cells feel when they're in the International Space Station, and also in 1/6 of the Earth gravity, that is the one that you can feel when you're at the moon, and 1/3 of the Earth gravities when you are in Mars. So we were using again Candida albicans. That was the one that I used in later in the other investigations that I made. And another team was working at the same project, but they were looking for antimicrobial resistance and how it changed between the different gravitational regimes. So if you can see, the things that I was gonna make with Candida in this project were very unlike the ones that I already did in the other clinostat project that I did, with the slightly difference that I was not only comparing the control results and the microgravity or orbit cells, but I was also comparing to simulated lunar gravity and simulated Martian gravity. So I cannot put the specific data because as I told you, it is a NASA-funded project, and I am not allowed to give you like specific numbers of what I accomplished with this project, not until it is public. So I can show you like this very general graphic that I made with how it behaves and how it changes its growth phases when it is under simulated microgravities. So for example, in this line, the 1G, you can see in here, we have the first black phase, or the phase where bacteria, and in this case, Candida albicans, the yeast, is like being accommodating to the gravity and the growth medium and everything that is with the cells. And after that, the cells start their log phase or their exponential growth in here. And after that, we have this stationary phase. We didn't accomplish to get to the death of the cells phase, but we have the stationary phase. And every time we want to make a growth curve, we want to see where the exponential growth starts. So it is okay we didn't get to the death of the cells phase. So we are going to be comparing to, for example, simulated microgravity. You can see that, for example, exponential growth started a little bit later comparing to the one of the normal gravity. Right in here, you can see the differences of this curve in here and this curve in here. But we accomplished to get only to the log phase and not the stationary phase. It means that under simulated microgravity and maybe also under real microgravity, the Candida albicans cells are gonna keep on their log form or log phase for more time, having a bigger optical cell density at the end of our experiments. Then if we are comparing, for example, the lunar simulated gravity, we're gonna be looking at something pretty similar to the one that we have under normal gravity, with the only difference that we have bigger optical density, meaning that we have much more cells than we have in normal gravity. And under simulated Martian gravity, you can see that again, the log phase starts a little bit later, but we can accomplish to get into the stationary phase in here. And even we can see the depth of the cells in here. So getting this into mind, that you can make not only orbital simulated gravity with the Clinostat, but you can also simulate, for example, other gravities like Martian or lunar gravity, is pretty important because it helps to get more importance to the investigations that you are proposing when you are, for example, gonna propose for the Artemis project. So you can send something to the lunar orbit, or for example, to the Martian orbit with more investigations that are coming ahead. So you can, I don't know, like put more information on your proposals when you have this kind of simulations that you are going to be able to make. So it is important to have a Clinostat. And in conclusions, well, Clinostat can be cheap now. You can have your own if you have $108. It helps developing countries to start their participation on space sciences. As you can see, myself, I'm a pretty good example of how Clinostats by themselves can help a lot to have countries into the space radar. And it can be used either as orbital analogs or other gravity analogs. So with this, I finish my presentations. Thanks for your attention. And also I have some acknowledgements to Dr. Licea, Engineer Freddy España, PhD candidate Pamela Flores, and also Josh and the Science on the people that are the ones that helped me to get into these kind of projects. This is my contact information in case you want to send me a mail, in case you want to have my number, in case you want to talk about anything related to space. Feel free to reach out to me. And right now, I think we can have a moment for questions.
I have a question if I can start. I don't know if there's a moderator or not, but can hop in with a question. So I was curious, you said you were culturing Candida albicans, and whenever I've cultured them, they've been adherent. And yet most of your intro was talking about cultured in suspension. What are the main differences between culturing in a clinostat that's either adherent or in suspension?
Can you repeat the word that you used before suspension, please?
Adherent.
Oh, mainly the morphological changes, because, for example, as you could see when we were under gravitational regimes, and even if you are, for example, having your candida cultured on agar instead of broth, you can see that the cells are going to start doing these morphological changes. When you have your cells on their agar plates, you're going to find cells in their Eiffel form because they're starting to be trying to get the most amount of nutrients that they're going to be using. And if you have them under a broth, something liquid, you can see that they are going to be in their budding form. So they are going to be circular. And the optical density is pretty obvious when you have these cultures, because at the first time you have the cultures in a translucent form, you are going to be looking at a broth that is very clear. And after some hours or even some minutes, if you want to wait like half an hour, you're going to be a huge difference. You're going to look a huge difference on the turbidity of the medium. I don't know if that answers your question.
I can actually rephrase it in a much simpler way. Can you culture adherent cells in a clinostat?
You
Does
can.
it make sense?
Yeah, you can. But it is not like the ideal form that you want to use them. Because, for example, what I have seen is that when people want to make that kind of cultures, they use the random positioning machine. Because as you're going to have the cells inherent to something, then what you want to cause to the cells is that they lose this gravitational location capacity. So if you want to have them under simulated microgravity, as they are going to be feeling under the International Space Station, you're going to use the clinostat for a constant freefall. If you're going to use it for some of the qualities that microgravity in the International Space Station has, for example, if you want to study biofilms or communities of cells, you're going to use the random positioning machine, because those are different principles for the cells.
Thank you. That answers my question. Thank you.
Okay.
That was an excellent presentation, Katherinne. Yeah, I think along the lines of what Kyle was alluding to as well, part of the challenge with clinostats might also be mimicking the in vivo cellular environment and the behavior in the experiments. So curious to hear your thoughts there, and if we could somehow coat some thin biocompatible coating on the interior, or yeah.
Can you repeat that question? If you could use what?
Some kind of biocompatible coating on the interior of the clinostats, and kind of help design some more in vivo experiments.
Yeah, for example, there are some experiments that have been done. For example, as I told you, one of the problems of the cultures that I made is that Candida albicans needs to be cultured under aerobic conditions, and I was making them under anaerobic conditions. For this, there are solutions of cellulose membrane that they use, so they can have a gas exchange with the medium that you're going to be using. So you can use that if you want to make it like more in vivo in that way. Also, there are other investigations that use certain coupons that are called, that they cut like small pieces of material that they need to look for, and they put it into the containers that they are using, so they can see how the cells behave in these conditions. One of the things or the problems that you have if you want to introduce something with this kind of cultures is that you need to make it anaerobic. Because if you use aerobic conditions and you want to see the interaction between the cells that you are investigating and the surface or the other kind of cells that you want to investigate, the cells that are pathogenic tend to prefer the cellulose membrane that you're using for gas exchange instead of the cells that you are trying to investigate. So what you are going to be using, or if you want to make that, I suggest that you use bacteria or microorganisms or cells that can be grown under anaerobic conditions. But yes, it can be done. I don't know if that answers your question, just to be sure.
Yeah, generally. Yeah.
Okay. Christine, I think you had a question too, right?
Yeah, that's right. So I have two questions. The first one is more clinical related, and the other one is more mechanical or hardware related. So with regard to the clinical question, so I'm presuming that since astronauts or perhaps people who have changed their usual gravity environment, they are now under stress. So probably their bodies are producing more glucose. So I'm wondering if there's a combination of factors there, why astronauts might be more predisposed for candida infections. The first one being because of increased stress, and the other one because of the microgravity environment. Would that be accurate to say?
Yes. Well, I'm gonna divide the answer. First, I'm gonna start with the glucose expression that is encased in humans. There is a study of twins that they made so they can see what happens to humans when they're too much time on space, and they didn't find like an enhancement of glucose expression. The thing that happens in the body of the people that are in space are mechanical related also and biological related. So for example, in the biological relations that people suffer when they are under microgravity conditions, let's say the astronauts or people that change the gravitational regime that they are in, is that the immune system is decreased when they are under this gravitational regime. So for example, some of the things that changes are the expression of some of the immunological cells that they express when they are under microgravity, and also the fluid dynamics that the body has is different. So for example, when you are in the International Space Station, as you are constantly free falling, the liquids that are in your body are going to tend to stay from your waist to the upper face to the upper part of your body. So for example, if you have an infection on either your genitals or in your legs, if you have a cut and for example your hands, then even if the immune system creates the cells that it needs to protect you from these kind of infections, the cells are not going to be able to get into that place as good as they do when they are under the normal gravity that we have on Earth. So the immune cells that your body creates are going to stay in this part of here, and in your extremities or even your genitals, they're not going to receive those immune cells. So it is like I told you that this mechanical stress that they have and why the fluid dynamics don't work the same for people in space for like it works for us, and also the biological less or decreased expression of immune cells when they are up there. So let's compare, for example, the immune system of an astronaut up there to that of a child, someone that doesn't have like that amount of cells that can protect them from the bacteria or the stuff that are around us. And that is the problem, and that is why they suffer so much from the microorganisms that are up there. I don't know if that answers your question.
Yeah, interesting, because the first thing that came to mind when I saw the use of Candida is, I thought, you know, Candida thrive more in glucose-rich environments.
Mm-hmm.
So that's what came to mind. I thought, perhaps because they are more under stress, so their bodies are producing more glucose, and maybe they have more fungal infections than normal. So my other question is about the Clinostat. Do you also perform bench testing? Do you keep the machine running to see for long periods of time to see if any failures occur?
Can you repeat? Sorry.
Yeah. Do you keep the machine running on its own?
Clip? Did you say?
Yeah. Do you keep? Do you allow the machine to run?
Mm-hmm.
Without microorganisms, perhaps, not just for testing, just to see if any potential mechanical failures, hardware failures, present themselves, so that you are able to see, you know, from a quality related perspective, to see what can, if it is used for a very long time.
Yeah, we made that, and something that was worrying us was that, for example, some cultures are made under 30 degrees Celsius, but other ones are made in 42, 45 degrees Celsius. So we're very worried about the temperature changes also, because we were using like elastic bands and stuff. So in the first Clinostat that we made, that strange stuff that was just like something round with more round stuff, and we were trusting gravity for this. We didn't have that problem because the motor that we had was protected by this cage that we were using, so it could run like for hours. We put it for 3 days actually without any samples, like no bacteria, no nothing, just water into a 15 milliliters conical tube, so we can see, for example, if the motor lower the velocity that it was working on and stuff, and it didn't. But for example, for the second iteration that we made, the vibrations of all the hardware that we were using were so big and so strong that everything was falling apart. So we were having the grains like working pretty good in the first day, and in the second everything was falling. So the things were not the dense of the grains that we were using didn't fit anymore. So we had that problem, and that is why we didn't make a verification test. And for the third one, the problem that we had was the elastic bands that were used in there tend to get too dry, so they broke every time we left it. We leave the Clinostat for too long, so we changed the material of the elastic bands that we were using in order to keep that problem away. But yeah, we made some verification tests without bacteria, so we could know, for example, that if we started the experiment now and we wanted it to be like 3 days long, in 3 days, I'm not gonna be coming and finding out that my experiment failed and I waited 3 days for nothing. So yeah, we made that kind of experiments.
Okay. Good, something useful to include in the instructions for use or the manual.
Yeah.
Thank you.
Check your temperature.
Thanks.
Thank you, Kristen. Any other question?
Yeah, I had a few questions here as well. It was reading through your awesome study here, and I think it's really important research you all are doing, and I part of me believes that the Candida fungus is one of the largest underrated outbreaks in the early 21st century. And I've personally had a Candida infection for the past 20 years. I think it's partially genetic, and I think realizing now it could be somewhat of a guinea pig in for you or others and researchers and possibly in microgravity. But you may also consider the specific strains Lusitanae, Cruce, and Glabrata. I think those are some gnarly ones there. So.
Yeah, I wanted to use more strains, but I was undergrad and like my mentors didn't trust me that much to give me that kind of strains. Also, when I was working with Candida albicans, I myself got an infection of Candida albicans, so they were like, no, we're not gonna give you like any other strain that already you have. So yeah, it is pretty important. Also, the ones that I use are the ones that are most commonly found on astronauts when they are under microgravity conditions. So, for example, for Apollo 14, I think, they made an investigation taking all the samples of, for example, mouth, genitals, blood, feces, and urine of the astronauts before they went to space. And when they were in space, they took the same samples, and when they came back, they took again the same samples, and they found out that Candida albicans is one of the main problems, and one of the cells that like take advantage of the poor astronauts when they're under microgravity conditions. So that is why I focused on Candida albicans. But yes, absolutely, we need to have more strains under microgravity conditions and simulated microgravity. And also, just so you can know, oregano oil, that is the one that I used for the first investigation that I used, demonstrated that it works to inhibit this yeast growth. So in case you want to use oregano oil or something more natural for the treatment of your Candida infection, let's say that you can use oregano oil even if you are on Earth on space. Just for fun fact.
Hi. First, I wanted to congratulate you on such an excellent sort of a low budget maker project and apparent success with your experiments. So I have, I guess, maybe a couple of comments. One of them is your use of for detecting the culture growth, and you're using the optical density of the culture. And I just wanted perhaps to suggest you could correlate that also, since the cells are apparently, at least in the zero gravity, changing from a spherical to a hyphal stage, and that might actually change the actual cell count response due to optical density. So perhaps you might want to do a sample and use something like a hematocytometer and do an actual cell count and a microscope to maybe correlate that, make sure that those growth curves are not being altered because of the morphology change of the cells. And then the second one was in the engineering. I know you said you weren't an engineer, but in the construction of your clinostat, you're actually using a stepper motor, and stepper motors are not exactly known for their smooth rotational ability. They step, and so I know you mentioned the problems you had with that one iteration of your clinostat with a vibration. And although you might not have a severe problem due to the stepper motor and using the microcontroller, the Arduino, in order to control the speed, I'm sure it's a lot easier with just in controlling the actual rotational speed of the stepper motor. It's much more, it's an easier process with just the software. But I might suggest you might want to look at changing the stepper motor to just a regular standard motor with a speed control on it, and then that way that would give you a smoother rotation of the system. So I'm not sure if that would cause a problem or not, but since you mentioned the possibility of vibration having an effect on the actual cell growth itself. So there's just 2 points that I wanted to, but congratulations, really excellent development work there. And best of luck on your future research.
Thank you. And yeah, absolutely. You're not the first one that tells me that morphological changes can also affect the density. So yeah, thank you for telling me, because I had already forgotten the things that other people told me about that. So absolutely, I'm gonna take it into account for the future verification tests that I make. And also for the stepper, that is something that we want to change in the open Clino, that we wanted to change the stepper because of those problems, but we didn't know like what kind of motors could we use. So if you can give me like, you already gave me the name, but if you have, for example, I don't know what brands can we use or some more specifications, I will be pretty happy to have your information. So thank you a lot.
Sure. Well, a standard motor does not have the discrete steps. It has a smoother rotation. So there's a lot of small motors, some that are, for example, used on hobby robots, and they're geared, so you can get them with a whole series of rotational speeds per minute. So they already have a particular ranges. You don't need very high speed for Clinostat. So let's see. There's a lot of electronic companies. If you just kind of do a Google search for hobby robotic motors that are geared, you'll find a whole set of those from a lot of different companies that sell those. And then some of those motors have actually a detector built into the motor that'll give you an output that correlates to the rotational speed of the motor. And then you can just use software also from, you can still use the Arduino, and then through software, you can actually detect then the actual RPMs, the rotational speed of the motor, so that you can control and maintain your constant speed. That of course you would need that, and or change the speed of the motor if you want to change, for example, your lunar versus Mars versus zero gravity speeds that you would want to rotate the Clinostat at. And so that could easily be accomplished by just software control. So just do a Google search for hobby robot motors that are geared and then have the rotation sensors on them. I wish I could just off the top of my head that I could think of a company, but I'm sure if you do a Google search through some of the maker sites, where people build robots and computer controlled robots and so forth, you'll run across a whole series of these, and they're fairly inexpensive. They're not going to be they might actually even be a little bit cheaper than your stepper, but in the same price range. There won't be a huge increase in cost. So the only thing that might be needed to change is the actual driver for the motor. I'm not sure if you could use the same driver that you use in the board that controls the stepper from the Arduino. It might require a slightly separate driver board, but again, they're all in the same price range that you're looking at. So sorry, I couldn't be any more specific.
No, but the specifications that you gave me are perfect. So thank you.
Okay, well, I hope it helps. And
Yeah,
again,
thank you.
that's just wonderful work. Congratulations.
Thank you.
Are there other questions or comments?
Sure. A thought reading through your paper as well. Wonder how we could use these clinostats or random positioning machines for material testing and specifically more on the abiotic side or the non-biological testing in alternative gravity. Could we add some simulant inside one of the tubes or something and get some cooking going experiment?
I think that only if you're gonna use like nanotech or something pretty small, because for big stuff, for example, if you're MIT does some experiments that I really blows my mind. For example, they use some of the solutions that I told you, the parabolic flights, so they can test some technologies or some materials that they are interested in. And for example, they were trying like mechanical claws, so you can stick to stuff. So you can imagine like astronauts going into the International Space Station and using their superpowers with, I don't know, these claws that they were trying to use in this kind of stuff. And also, if, for example, you want to study, I don't know, fuels or some materials resistance to microgravity and stuff, like specific materials or specific characteristics of the things that you want to study, you can use the drop tower test. That is, as its name says, it's a huge tower with a huge thing that they drop from the top of the tower to the bottom, and you have like a minute or 30 seconds of microgravity in there. So people use it to see how the fuels work and how they can better the fuel capacity when they're under microgravity conditions, and a lot of stuff that are not related to biological experiments. So if that helps you with your mind cooking that you're doing.
Yes. So we're definitely interested in collaborating with you. You know, we'd love to help support and develop the research around other testing with microorganisms. You know, maybe some cyanobacteria and Anabina. You know, Shonella, and you know, maybe some spirulina with Terry Trevino here if he's still on. Yes.
Yeah, I will be happy to collaborate.
Awesome. Hey, you've inspired me to take a deeper dive into the Candida too. So, yeah, we're also interested in, you know, potentially designing and building some clinostats during our MarsU programs, and you know, if you'd be interested in advising some students or something sometime. Really cool.
Yeah, I'll be happy. And you have the design there, so you can print as much Clinostats as you want to, and I can help you with the designing of some projects and stuff. So yeah, feel free. And as I told you, I'm pretty happy to help. So it will be an honor to me. I don't know if you have any other question, AZ. You're muted, I think.
Okay, can you hear me now?
Yeah.
Okay, I don't know if you have access to the chat. I put a link there to a supplier that provides those motors, and if you do have access to the chat, there's the link on it.
Yeah,
if
thank
not,
you.
if you see that, then they have a whole range of those motors, and then the drivers, everything you would need for altering that design.
Thank you.
So, okay, you're welcome. Bye-bye.
Yeah, thank you. It's gonna be very useful. I don't know if there's any other question. If not, I think I saw a lot of you were following me on social media. So if you want to make more questions in social media, feel free. If you want to make some more questions in here, also feel free.
Thanks so much, Katherinne. We'll be in touch, yes, and for everyone for tuning in this month.
Thank you, Katherinne, and goodbye, everyone.
Goodbye. Nice to meet you all. And thank you for your attention.